Okay. Yeah. Hello, everyone. Welcome to the MIF++ seminar. It's a pleasure to introduce Laura Gardiner, who will talk about harnessing multi-omics foundation models to accelerate biomedical discovery, so delivered from IBM Research, UK. Over to you, Laura, please.
Thank you. I've probably not instilled much confidence now. You're like, she can't even join teams, man. What does she know? But, you know, I assure you, this will be interesting, right? So hi, nice to see you all. And I'll just get started. I mean, just to give you a tiny bit of background on me, I'm actually a biologist by training who went into, a wet lab scientist. Biochemistry was my background. I went into computational biology for many years. And then for the last 8 years, I've been at IBM Research applying AI in that space. So mainly to multi-omic data, to address kind of biological problems. And I'll tell you just a little bit about us because it kind of leads into the research we do. You might have heard some from one of my colleagues, Dave, in the past. But at the moment, I'm based in Daresbury, in the north of England. So obviously quite near to you. And we're part of a collaborative centre with STFC. So we're at the Hartree Centre on Daresbury SciTech Park. And so IBM, STFC sit hand in hand, and we've been part of a 5-year programme where we work together. And the idea, it was kind of partially government funded, partially IBM funded. And the idea was to essentially enable UK businesses to adopt AI in a safe environment. AI and quantum, but I'm mainly focused on AI, so that kind of bias is what I'm talking about today. So it's a 5-year program, so we do a lot of projects where it'll be IBM, STFC, who are kind of the government partner, and a client. That is how I would describe them. So a third party, normally an industrial collaborator, could be a university. We've done some work with the Earlham Institute, so a few institutions, Rothamsted Research, but mainly clients, I'd say, from the kind of pharma industry, that kind of thing, SMEs as well, and some government bodies as well. So always us, SDFC, and a third party on a project. So the team I'm part of, on site in Daresbury mainly. A couple of people have kind of recently moved down to London, but there's a small group of us who are dedicated to healthcare life science and a much broader team generally at Daresbury. But obviously we're very connected to the IBM global team. So there's kind of thousands of us all together. So there's a really wide network. But this is my team. They're all fab, and our expertise is AI, bioinformatics, and software engineering. And our mission is kind of guided by the broader IBM strategy. So we're accelerating primarily drug discovery and enabling precision medicine with generative AI and quantum. As I said, I'm more on the AI side, so you're going to get a complete bias to AI today. But doing things like understanding disease mechanisms, stages and subtypes, early stage target discovery, and designing new small molecules and biologics, and predicting patient-specific drug response. So typically when we're looking for clients or collaborators, we're kind of targeting people working in that space or with data that might kind of relate to that. This is things you already know, but I think it kind of lays the foundation for what we've been doing, this kind of walkthrough of where we've been. Obviously IBM's slightly archaic as a company, but we've kind of followed this trajectory through from rule-based systems to machine learning, which we still do actually a lot of, to deep learning, which we still do, I'd say, a fair amount of. And at the moment, we're more focused on what we've called foundation models. So, these large generalized models. And I'll talk a bit more about kind of them in our context. So our context is kind of DNA, RNA, protein, language mainly. So we're looking at this paradigm, which we've been stuck in for a long time, where we've been building these small models based on, you know, cohorts or data sets that were quite limited and small. And our models have been limited, I guess, to the universe of the problem task and the training data. And, you know, it's a massive problem with generalizability, how to lift a model into even a new cohort from the same disease. You know, we've historically struggled with training data, particularly labeled training data, generating models with these huge feature sets. So we've had many high dimensionality problems where we have lots of features and not many patients. And then we've entered this kind of foundation model paradigm, which got us really excited, I guess, as biologists, where we could see this might be of benefit kind of fundamentally, where you're having these large models trained on huge unlabeled data sets, which we have. We have lots of data sets that might not be labeled for our tasks, say some rare disease or a cancer type. So the idea being a model might learn generally across multiple tasks and domains, and then we would fine tune them for our specific task. Let's say it's chemotherapy response prediction or something like that. And we can have these models much more easily be multimodal. And you can see how this might appeal to us when our main problem was small data sets and a lack of training data. But what I wanted to kind of show this is because this was never natural to me, because I could see the large language models like ChatGPT or whatever it is you're using, and I could understand how they could train huge unlabeled, you know, self-supervised models. That made sense to me from the language point of view. You might mask, you know, a word of a sentence and try and learn it. But I was, how's this going to work for DNA and RNA? And the main approaches that we are using are, you know, very similar. You know, we're treating our DNA as a sentence, just like I think you might see for protein or for natural language, and, you know, masking elements of it. And with our RNA, that's typically what we're doing is using count data, so gene expression levels, things like that. And we're masking gene names or expression counts to train these models. So this way we actually have a lot of data in the public domain. It's just that it might not have been labeled for a specific disease or tissue type, for example. So that's what I mean when I'm saying we're kind of generating models with unlabeled data sets. And for us, this is kind of becoming, you know, the obvious next thing, because you know, we can pre-train on these large data sets that we can kind of scrape from public databases. You know, we might have millions of samples that are unlabeled. We might learn the language, you know, of RNA expression, of DNA, and then we might tune a model for a specific disease or task, fine tune, and then we're looking at thousands of samples, which is much more tractable than working with a client or a partner. And we can kind of get those numbers relatively readily. And then you have this kind of possibility for inference downstream when you've trained your mod, when you fine-tuned your model, where you only need very small numbers of samples. And this is also something that happens to us a lot, you know, where we might have less than 100 samples. So that's kind of what we're calling inference and downstream tasks. And obviously the overall idea being here that once you've learned that base language with your pre-trained model, we can get a performance gain with only a small amount of labeled data. But what I'm going to talk to you today about really is, you know, we say this a lot, but we wanted to prove it. So we actually want to use our models in real applications with real clients. Do they actually improve upon the baseline? Kind of put your money where your mouth is time. We see a lot of benchmarks, but do they really hold up when you've got real data and a real problem? So I'll kind of just go over a couple of our models, and then I'll show you a couple of those applications. So shamelessly plugging our models here, but here's our models. So they follow the the trends that are around at the moment. The two top ones are not ones I've been involved in, but I will talk about them. They're probably our most mature, I would say, that they've been around longest. I think they're both published now, finally. So we have melon and mammal, and you can see that the graph at the top kind of shows you the distinction there, where they're both kind of, one, melon is for small molecules, mammal for biologics, but melon spans kind of different types of data views. So, sequence, image, graph, 3D structures, whereas mammal is trained on different data domains, really. So small molecules, protein, RNA, DNA. And melon, we fine-tune that for things like predicting toxicity of small molecules or metabolism. And then with mammal, you're looking at more things like predicting drug protein binding affinity. And I'll show an example of us actually applying mammal for some advantage in a minute. But the other ones, obviously, that you might guess are more close to my heart, we have our biomedical foundation models in RNA and DNA, and they were more along the lines of target discovery for their development. So integrating large RNA data sets, fine-tuned for things like cell type classification, gene perturbation analysis, drug response prediction, and our DNA models for things like promoter detection and SNP disease association. Let me just show you. I think I'm going to have another image on that, and then I will just show our kind of overall stack as well. So the idea here being, as you've hopefully gathered, that we have multimodal data going into a pre-trained model, and we want to enable downstream tasks like gene disease association. We're doing antibody property prediction, antibody design, and mainly target identification and things like that. I do have some real examples I can talk about, don't worry. But this is, so actually not to be forgotten, is our whole stack. So I think we get a bit obsessed with whatever we're working on at the moment. And I thought it was important to show this because we very much use all of these tools still. So our kind of bioinformatics can't be forgotten. We work on genomics, transcriptomics, proteomics, metabolomics, microbiome, metagenomics and metatranscriptomics. So we kind of have all our workflows for those kind of data sets. We're very interested in kind of feature filtering, feature selection, augmentation, and engineering of features from these data sets, normalization and transformations. And then we have our kind of AI layer where we still very much revert back to classical machine learning as our baseline. I always get asked that, as you just put classical machine learning in the bin now. No, we haven't at all. And that's often our baseline. Deep learning, particularly graph neural networks, we still do a lot of. They're very competitive in some of the things we're looking at. Foundation models are something we're quite excited about at the moment, and all our foundation models are open source, so you can check them out. And of course, quantum is kind of where we're hoping as a company, the future is. That's where IBM's betting. So we're more and more looking into how that might be able to help us. Then that kind of last layer we have is the kind of explainability interpretation. And you can imagine if we're working with clients, say, I don't know, life science industries, they are probably more interested in the explanation and the potential biological mechanism behind what we're predicting, or the potential, predictive genes or whatever it might be. That's often actually more exciting to them than the actual performance of the model, presuming that the model performs well. So it's a kind of something we're not blind to at all, and we've got a lot of energy in that area too. So that's our kind of whole stack, and then I will take you through some applications that have been driven by the team. So the first one is using mammal, which is more in the protein context for inference of drug target binding affinity. But what we wanted to do here was we were working with a partner called Reprocell, who were very interested in IBD. They wanted to find novel targets, and they wanted to know why patients respond to drug treatment differently. So they have small numbers of patients. They take diseased tissue from the colon. They do transcriptomics, genomics from that. And their predictive target is actually a proprietary assay from themselves, which measures kind of what they're taking as a proxy for drug response. And that they kind of gave us treatments from a selection of drugs. Here I'm going to show prednisolone. So they're trying to predict patient drug response, essentially, whether they have a good or a bad response from the tissue, transcriptomics and genomics. So what they were very interested in was they actually had a very robust workflow already, where they were using kind of classic machine learning. And we wanted to think outside the box for them, like how can we use these models? They didn't want GPU, they didn't want too much computational expense. That was another thing. So what could we use out of the box for them to improve what they already have? That was kind of the question. And at the time, that was quite a big ask, because not many of the models were performing that well at the time. So what we looked at was what we've called kind of feature selection and engineering using the foundation models. So accepting their workflow for what it was and looking upstream. So saying, how could we use these foundation models to actually generate them some new insights, new features? So they've got genomics, they've got transcriptomics. What we were looking at was using the genomic data, the patient SNPs, to evolve, those that maybe impacted protein sequence, to kind of mutate a protein to make a patient version of that protein and calculate patient-specific protein drug binding affinities. So I'll talk a little bit about this in the next slide, but this worked well for us. We were able to, detect known targets and identify new biologically coherent targets for the drug. And, we were able to improve upon, obviously, the classic machine learning approach that they had already. I'm kind of skipping to the end. I shouldn't tell you the end, but there you go. So this is what we were doing. So quite simple, really. We were using MAML to essentially rank and prioritize genes initially. So we took all the reference proteins from the human genome and their drug of interest, just prednisolone and SMILES format, and fired them through MAML. So completely data driven. We don't need to know anything really. We just calculate drug target binding affinity using inference from that model, and we assess the binding affinity of each protein to prednisolone. So we can do this just for the reference proteins, and we can rank them according to their binding affinity. We can select the top X%. We can use this to prioritize. We looked at some of the proteins in the top 5%, we were seeing obviously known targets right at the top, but we were also seeing other sensible enriched functions there, and potentially, since this is kind of a broad ranging drug, we were finding other sensible targets. So this is really useful as kind of a data driven, screening tool. Just using inference from a model that's already there. We didn't need GPU for that, so that kind of hit their expectations of kind of computational frugality, if you will. And then stage 2, we wanted to actually evolve or mutate the reference proteins to make them kind of patient specific. So we're using the SNPs that Replicell had, altering the proteins, mutating the proteins, if you will. So you've got a patient-specific set, and then you're recalculating drug binding affinity and kind of calculating a new binding affinity, really comparing the patient binding affinity to the reference. Do you know what there's like? Sorry if you can hear all that noise outside. There's people washing the windows. They're waving at me. Okay. I'm just going to close the window. Sorry about that. Right, so we have these kind of what we're calling them kind of newly engineered features. So rather than using what Replicell were using, which would have been their SNPs in a model, we're now kind of developing features from them and using them to calculate patient-specific binding affinities at the gene, and associated protein level. So we're also kind of, doing a sort of dimensionality reduction on the data because many SNPs might impact one gene. So the newly engineered features could be used for their classic downstream machine learning to predict drug response as they were doing. So it kind of seamlessly fit into their workflow. And we just compared up, like, did this actually have a benefit? So using like a standard feature selection, but compared with SNPs, using feature selection, using the kind of foundation model guided, so as in which were the most highly bound proteins to the target drug and which SNPs fell within them, and using them for prediction. So kind of we're calling it foundation model led feature selection. That improved on our predictions. And then the third feature set was kind of using these binding affinities instead of SNPs, so patient specific binding affinities, for the set of mutated proteins to predict patient drug response. And it worked really well. In each iteration, our predictions improved. Kind of giving confidence that you can use these models. I guess what we're trying to show is that you can use these models in ways they weren't necessarily intended to fit into your current workflow. And the part that the client were most interested in, I guess, was the explainability at the end. So here we're using quite a simple, classic machine learning approach ultimately to predict drug response. So we were able to just use SHAP to tell us what are our most predictive features. And we saw, a mixture. So at this point, we've got a best model where we've incorporated our binding affinities, our SNPs, we've got our transcriptomic data, and we found a lot of olfactory receptors coming up, which was quite sensible. Now we look at it and we've proven that they were expressed in the tissue, but quite actually novel for them to investigate some of these genes. So kind of exciting, really. But the workflow itself, I think, is what we were mainly developing for them, and they'll be reproducing that with their other data sets. Another kind of edge to it that we thought was quite cool, really, was that we did an MCP server of the mammal model itself. And just to kind of show the reproducibility element in that case, like previously we've been doing big sets of code and software to allow people to reproduce our workflows, but here, just using Claude, we were able to kind of feed the MCP server for mammal in and say, can you calculate binding affinities for me between the SMILE sequence and my FASTA files? And it wanted to and fed into kind of FASTA files, mutated and reference, and it's been able to just do that right off the bat really, really beautifully, quite embarrassingly so. Claude can do it in a couple of seconds, so you can kind of use these tools and perform model inference like that to kind of reproduce our work very easily using an agent, but just adding that kind of MCP element we found made it more reproducible and reliable. So that's what's happening here, and we do have a paper where you can access this code and get a few more details. But that's one application that hopefully shows that just because you've got a protein model and you've got genomics data doesn't mean there's a problem. You can still maybe help you. And our other application was more centered on RNA. So one of the biomedical foundation models that I mentioned, BMFM RNA. And here we were doing disease classification. So I'll move on to this one now. But again, hopefully trying to prove that these actually work. So this was a project that we did with Cleveland Clinic London, obviously us and STFC working together. And the idea was that they wanted to feed in patient RNA-seq data from specific states, let's say disease versus control. They wanted to compare, I think what a lot of people want to do, a series of foundation models against the baseline, which would just be TPM counts, normalized gene expression counts. So would embeddings from a foundation model improve upon the baseline for prediction of say, classification of disease state. And they want to identify genes differentiating those states and assess biological plausibility. So, your query will be your data. You'll select a range of foundation models. As we have our RNA one, but we have a load of others out there. There's GeneFormer, there's SC Foundation, SCGPT. There's all these models that we could use. Extract embeddings, train models, perform classification, and then we want to get explainability out and able to rank genes that were most predictive. So I think we had about 400 samples for this. Let me just skip ahead. So this was our workflow. And again, we are actually trying to make this agentic at the moment with Claude Code, I think we're going to be using. So you've got your data sets, you've pulled down from just Geo. These were actually public data sets, although they do have their own private ones, but we've demonstrated the concept publicly with public data. So then we're feeding each it's just a CSV really of normalized gene expression values into a series of foundation models, getting embeddings out. We are setting up quite a complex experimental setup for testing where we're comparing kind of within cohort and cross cohort training and testing, which was quite illuminating for us. So as in within cohort in this instance, samples from the same study, the same cohort, whereas cross cohort was that the model would never see in the test and training data anything from the same cohort. So probably a true test of generalization, if you will, because different sites, different studies, different times, completely different teams doing the work. Then we have a tool called Auto Explainable AI for Omics, which does kind of classical machine learning and SHAP explainability. So we're training in a series of models here and selecting the best. We did have a specific explainability for foundation model embeddings here because it's a mixture of classifier aligns, integrated gradients, so trying to use SHAP embedding, SHAP rankings of feature importance or predictivity aligned with integrated gradients essentially to try and get some interpretability even from the embedding point of view. And then obviously we were looking at knowledge graphs to kind of query the predictive genes. So that's our kind of broad workflow. But what we wanted to do mainly at section kind of 2 that I'm not really showing there is that we're using quite a few foundation models here and selecting the most performant later on. So our results, what we noticed was that across the board, the TPM count, so the vanilla, if you would, so no foundation models, we're training models from them and they're overfitting, and you see this vast performance drop, particularly when you changed across cohort predictions. And this was quite alarming for us, really. But almost all the foundation models we tested, when we used embeddings to train our models, they performed significantly better. And what I think was even more striking, so that's good. I mean, so they're working, they're capturing something. But what we also noticed was when we looked at explainability from the within cohort compared to cross cohort models, the same genes were predictive for the foundation model embeddings. So as in there's quite a lot of stability there, the same things are being used to predict, and the models, produce like pretty much as accurate performance. Whereas when we use raw TPM counts, not only was there a massive drop of performance, but there was also completely different gene lists between within cohort and cross cohort models, which is quite alarming when you're a biologist relying on that, for your work. So this is something we found really interesting. So we would have called, the foundation model embeddings were not only kind of increased performance, accuracy, and an F1 score, but there were also kind of stable, generalizable genes that were being pulled up as being most predictive, which gives you a lot more confidence when you're then doing your biological interpretation downstream. And we're identifying from, kind of new genes linked to, it was actually endometriosis was our disease of interest here, and kind of novel gene signature biomarker targets. Obviously all within pathways that were biologically sensible, that we knew to be involved in the disease, but we did get kind of new candidates coming up that were sensible. So this is really quite exciting for the client, and they'll kind of keep running the pathway. The workflow, sorry. So this is all available. As I said, we try to work with the client, and we try to not base our final tool or workflow on their data so that we're able to publish. So this was done with public data, and it's on archive at the moment. But hopefully, it's another example giving confidence that these are working in a real world setting. And I think what was maybe a little even more exciting was that we picked endometriosis because actually there's not much data on it. And a lot of these models have been trained on completely different data types. So we used a lot of bulk RNA, and some of the models here were single cell, and they work really well. And also they've not been trained on kind of any data from endometrial tissue. So it's amazing that they're actually improving performance nonetheless for our tasks. And that was mainly what I was going to say. So to kind of give you, hopefully I've given you an overview of us, that we're quite diverse in what we're doing in some ways, because we're applying it with loads of different clients, different problems. There's some agriculture problems in there as well, but our technologies have core similarities. So we're commonly working with kind of multi-omic data, disease linked problems, target analysis, but the idea being that we try to engage deeply with domain experts to try and bring the right technology and solve problems that are meaningful and really show that impact. So yeah, that's me. And yeah, as I did mention, all our models are out there. Our publications, they're not always the best performers in every situation, so we're quite open and honest about how they're performing. Yeah, and quite transparent really. So yeah, there's a website there that has all the models, all the papers, anything linked to them like that, and we have a Git repository, and they're all on Hugging Face as well. But yeah, I'm happy to take any questions or discuss anything really, if it's careers in industry or whatever it is. Happy to chat. But yeah, that's all I was going to say. Thank you.
Thank you, Laura. Please keep your slides. So first, let us thank Laura for the nice presentation. Now, usually I stop the recording, and after that, I invite questions. We have